and good night, depending on when you're watching this video. This is Mr. Messina here, ready to talk to you about solutions in diffusion as it applies to cells in biology. Now, a lot of students have trouble with this, mainly because they don't really know why this is really biology. This sounds more like a chemistry thing. Solutions are very important to understand because solutions are all very important to cells. Cell membranes themselves are semi-permeable. Cell membranes are semi-permeable, not cell walls. Those things don't allow anything through unless they're actually broken. Cell membranes are semi-permeable, which means water can pass in and out of them. It's not very easy, but it's not difficult at all for water to move from one end of a membrane to another. This means that a cell's survival is dependent on keeping the perfect amount of water inside of it. Too much water and the cell will literally explode. Not enough water and the cell will fail to function because it doesn't have the right ions. What's this balancing act, this keeping itself the same idea? That would be none other than our old friend homeostasis from Characteristics of Life. So, in order to talk about cell membrane transport and other things later in the course, we need to know about solutions, which means we need to know what exactly a solution is. Like I said before, cells exist in solution. And a solution is nothing more than a substance dissolved in a liquid. And this substance and these liquids have very special names. Ours, for example, the solution we're going to talk about is Kool-Aid. Now, talking about the liquid that we dissolve Kool-Aid in to make it actually Kool-Aid, it's called the solvent. It is the liquid base of whatever solution we're talking about. In Kool-Aid, you typically mix the Kool-Aid mix with water itself. So water is our solvent in Kool-Aid. Now, the substance that we're talking about in our Kool-Aid, that's called the solute. It is the substance dissolved in the liquid. Now, for Kool-Aid, our solute is going to be some kind of sugar or Kool-Aid mix because you want to actually be drinking the deliciousness of sugar instead of the Kool-Aid. It's instead of just some kind of like red, weird water. Now, water concentration obeys something called osmosis. That means how water flows from one side of a membrane to another. Now, there's three different types of water concentration that we need to be aware of. You have hypertonic, hyper, which this prefix right here means more. Hypertonic means there's more solute and not a lot of solvent. So a hypertonic solution you can see right over here. There's a lot more of those little dots than there are on the left-hand side. Hyper, more solute. Now the hypotonic right here, you can see that prefix. That means there's less solute. So that's our situation right over here. Right there. That means hypo, that there's less solute hanging around in the same amount of solvent. And then an isotonic solution actually means there's an equal amount. You have an equal number of solute on one side and the other. Now, water, the direction that water flows, it'll go from hypo to hyper. Isotonic, which is this right here, that's water's goal. It wants an even amount of solute moving through it at any given point in time. You can see that right here by this little arrow. Now, we talked about osmosis, but in addition to osmosis, there's another force that acts called the concentration gradient. Now, the larger the difference between one side and another, so for example, this is a very high concentration, while this over here has a very low concentration, these things over here do not like to be around each other. These solute molecules, and they're going to head this way. So not only is the water going from a high amount of water to a lower amount of water, but also the solute is going from a high concentration of solute to a low concentration of solute. So that means a high amount of sugar in your Kool-Aid mixture is going to try and go to a lower concentrated area. Same thing with the water in the Kool-Aid. The higher location of water is going to try and flow to the lower concentration of water. Now this brings us to actual diffusion. So whenever you have a semi-permeable membrane, you have this flow of high concentration to low concentration, just like diffusion. This is called passive transport. So if you have a membrane, which we're going to get into more details, so you have a little phospholipid membrane, and there's a lot of little particles out here, a lot of little particles out here. They're all trying to cross this membrane to go through here, because there's only one little thing over here. There's so many more circles on top, they're all trying to get to the bottom. This is called passive transport because it requires no energy whatsoever. It will just happen. Doesn't need a catalyst, doesn't need anything fancy. It'll just diffuse its way through. So osmosis is just the diffusion of water. So water is going to go from high concentration to low concentration just like a substance. So if we look over here, this one right here has a high amount of water, where this one over here has a high amount of sugar. Okay? This little dotted line right here is a membrane. That means the sugar cannot cross this way. Sugar is trying to go that way. It can't do it. It's too big. It can't cross that membrane. The water, on the other hand, these little blue pellets, they can totally cross. So the water is going to push over to the left-hand side, as you can see by these arrows here. And you're going to get this. This is what you're going to get. So the blue water molecules moved in this direction, and now you can see on the left-hand side there's a lot more water molecules along with the sugar. The sugar didn't go to the right. Sugar did not do that. And the reason that it couldn't do it is the membrane that separates them. This actually happens in cells as well. So remember, diffusion of water moves from high concentration of water to a low concentration of water. Not a low concentration of water to a high concentration of water. So, and as I said before, you have a semi-permeable membrane that's separating these two. That's this dotted line right here. In a phospholipid bilayer, which is actually what a cell membrane is, it looks like this. You can see this is the inside of the cell. I'll call this cell. We've learned before that this is called cytosol, and this is the outside. I'm going to just call this out. And you can see all the blue water molecules hanging out and all the big green sugars, ions, proteins, whatever they are, being separated out of the membrane. Now, this actually works more easily than you think. Slugs, for example. Their skin is semi-permeable, and you might have heard that adding salt to the outside of a slug causes them to shrivel up. So what actually happens is when you pour salt on a slug, since it's a semi-permeable membrane, this actually draws the water right out of the slug. So what normally happens when you put a salty slug in fresh water? What would you actually think would happen? I'll give you a second. That would actually, if you put a salty slug in fresh water, it would actually take the water off of the slug, and water would flow inside the slug and recover it. As crazy as that sounds. So in this particular situation, take a look. Inside the cell, we have a high concentration solution of 20% salt, and outside the cell, we have a lower concentration of salt. So what direction would this go? What would actually happen in this situation? I'll give you a few minutes to think about it as well. That's the end of this particular presentation. We talked about what is a solution. We talked about osmosis. We also talked about concentration gradients and how they would move. Tune in next time so we can talk about the cell membrane and how it actually is made up. This is Mr. Messina signing off. Have a wonderful day.